You're welcome everyone at Medical Sciences, where today we want to discuss about anti-human globulin test, what we call Coombs test. And this anti-globulin test, or what we can call Coombs test, was discovered in 1945. This one was discovered in 1945 by a man called Dr. Coombs, and that's why it is called Coombs test. So this Dr. Coomb and race, they discovered this anti-human globulin test. And this test, or this what we are calling anti-human globulin test, it uses anti-human globulin reagent. And this reagent was got by injecting. It is got by injecting human globulins to animals. So when you get human globulins and are injected to animals, so it is got by injecting these to animals like rats, mice, and rabbits. Then later, we collect the polyclonal antibodies manufactured by these animals after injecting them with human globulins. And that's why the name is called anti-globulin test, or what we call anti-human globulins. Antibodies developed against human globulins. So that's why it is called anti-human globulins. So after we have injected these human globulins to the animals, and then they are monitored, then we collect their serum or their fluid, we are able to get what we call either monoclonal antibodies or polyclonal. Or they can be monospecific or polyspecific antibodies. These antibodies, they are the one we are using to detect or to do tests which we are calling Coombs test. And what is its significance or the purpose? After seeing how they are produced, you can ask yourself, what is the purpose? The purpose of Coombs reagent, or what we are calling a human globulin test, this one is used to detect. Incomplete antibodies. This is especially IgG type, that is immunoglobulin gamma, or they can be used to detect the complement. So these incomplete antibodies, or the IgG antibodies, we call them incomplete because they do not cause visible clamping. They only cause sensitization, or they coat RBCs without causing visible agglutination, and that's why they are called incomplete. This one is different from naturally occurring immunoglobulins like IgM. For IgM is a complete because it causes visible clamping on reaction. But we are saying IgG because they are of small size. They are small in size. They are able to cause only sensitization. That if this is an RBC, if you have a red cell and you have these incomplete antibodies or the complements, especially complement 3, they come and coat. They only coat the surface of red cells without causing visible clamping or visible agglutination. So this is what we are calling sensitization. They sensitize the surface of RBCs. These are RBCs without causing visible clamping because they are small in size. So we are seeing this anti-human globulin, when added to these incomplete of the complement, they cause the visible agglutination by causing cross-linkage. So if I add anti-human globulin reagent, it causes visible clamping or agglutination by cross-linking these incomplete antibodies or the complement, hence causing visible agglutination. So this one results into visible clamping. So this is the purpose of Coombs reagent. Hope you understand it. it is used to detect or to diagnose presence of incomplete antibodies, especially IgG type or the complement within the body. These incomplete antibodies can be produced within the body, that is in vivo, or it can be detected in vitro. In vivo or in vitro. In vivo means the red cells are sensitized by these incomplete or the complement within the body. And here, if the sensitization is in vivo, we are going to see different forms. This one we do what we call direct Coombs test, what we call that. But if the sensitization is done in vitro, that is when you do it in a test tube, you get the serum from the recipient, then you combine it with the donor cells. We are able to sensitize these cells in vitro, and here we do what we call indirect Coombs test. So we are seeing that the different forms, we are looking at types of anti-human globulin test. They are of two forms, as we have seen. If it is in vitro, we are seeing we can do direct Coombs, direct anti-human globulin test, or we can do indirect anti-human globulin test. This one is abbreviated as DAT, and this is abbreviated as IAT. So these are the different two forms of Coombs test, and we are going to see one by one how they come about. But in summary, we are going to see that the direct Coombs test, this is a one-stage or one-step test, whereby direct Coombs, it is used to detect or to demonstrate already sensitized. This one demonstrates already sensitized cells, already sensitized or coated RBCs with either immunoglobulin, that is anti-IgG, or complement 3. So it is used to demonstrate already sensitized RBCs in vivo, that is inside the body. So in this test, we only collect blood, then we add anti-human globulin to demonstrate these already sensitized cells within the body. That is what we are calling direct Coombs. And under direct Coombs, we are going to say under direct Coombs that if I draw blood, that is blood from a patient, and this blood, I prepare what we call 5% RBC washed cells. And these cells, we are saying they contain, or they are sensitized. So if I have these different RBCs, which I have collected from a sample of the patient, these red cells are already sensitized with, immunoglobulins, that is IgG or complement. So these already sensitized cells, so I have RBC already sensitized. This is already sensitized with IgG or the complement. I have another one which is already sensitized. Another one is, so these cells are sensitized, but there is no visible clamping because they are of small type. I have another sensitized. So these are what we call sensitized red cells from the patient. And these cells, as we said, they are sensitized in vivo or in the body. So what we do, we add what we call anti-human globulin reagent. This anti-human globulin reagent contains the antibodies against the human globulins. So we add these antibodies against human globulins. When we add We add these ones and intubate at 37. And after intubating, we are going to do what we call centrifuge. We centrifuge at 1,000 RPM, that is revolutions per minute, for 1 minute. Then afterwards, if these red cells are already sensitized, they are going to cause what we call visible clamping. So we had our cells, our red cells, which were already sensitized. If they are already sensitized with these immunoglobulins, which is IgG or the complement, they are already sensitized. On addition of the Coombs reagent, these anti-human globulins are going to cross-link, or they are going to cause what we call cross-linkage of these incomplete antibodies. So it causes cross-linkage at the FC portion. It attaches on the FC portion of the incomplete antibodies. And this one, if I have another coated antibody here, I have IgG of the complement, here still it causes cross-linking. So this cross-linkage, due to addition of anti-human globulin reagent containing these antibodies, they produce cross-linkage or lattice formation. And this cross-linkage is the one that results into visible agglutination. So this is the principle behind direct Coombs, whereby in summary we have said they detect already coated red cells in vivo. And this one is demonstrated by adding anti-human globulins, which creates cross-linkage between different red cells, resulting into what we call visible clamping. So cross-linkage by anti-human globulin produces what we call visible clamping or visible agglutination. And this visible agglutination, as we are going to see in the procedure, it is demonstrated either macroscopically or microscopically. Either you tilt to see agglutination or hemolysis, or you do what we call on a microscope. So the procedure, but before we look at the procedure, what is the significance, or when do we apply direct Coombs test? This direct Coombs test, which you are calling DAT, it is used to demonstrate many things, many tests. For example, it is used to rule out hemolytic disease of the newborn. It can be used to demonstrate or to detect autoimmune disorders. Autoimmune disorders or autoimmune hemolytic anemias. It can be used to detect blood transfusion reactions, blood transfusion incompatibility reactions. So if someone is given a wrong blood group, which results into blood transfusion reactions, they can be demonstrated by doing direct Coombs. And lastly, we can also do direct Coombs to rule out or to demonstrate drug-induced hemolytic anemias, or what we call hemolysis. So these are the 4 applications of direct Coombs, which demonstrates incomplete, that is IgG, and the complement, which cannot produce visible clamping. They sensitize without producing visible clamping. So to see the agglutination or the clamping, we add anti-human globulin, whereby this anti-human globulin produces cross-linkage in between red cells, resulting into visible clamping, and we are able to demonstrate these diseases: hemolytic disease of the newborn, autoimmune reactions, blood transfusion reactions, and drug-induced hemolytic anemia. So those are the 4 applications of direct Coombs. But what is the procedure? The procedure of this test, you are going to assemble the requirements. And among the requirements we need is the centrifuge. We need washed cells. We need tubes. we need controls. Controls, we shall need positive and negative control. Those ones shall be needed. So after assembling your requirements, including 0.9% normal saline, after assembling these requirements, what you're going to do, after collecting a sample, either in EDTA or in a plain top, if I have a tube, we are saying we have a tube for the test, tube for the positive control, and tube for the negative control. So after assembling these tubes, we prepare 5% red cells. But before preparing, we first wash. So here we assemble, then we label tubes. Test, positive control and negative control. Then afterwards, we wash cells, red cells, at least 3 times using normal saline, 0.9% normal saline. After washing these cells, we prepare. We go ahead and prepare 5% RBC cells. And how do we prepare it? By getting 1 drop of cells, washed cells, you add them to 19 drops of normal, you add 19 drops of normal saline. So this is how you prepare 5% red cell suspension. After preparing, we are going to get 2 drops. You come and put 2 drops of 5% red cell into the tube for the test. Then for the positive control, you put anti-human anti-D globulins. Then here you put normal saline. Negative control can be normal saline. Then here you put anti-human globulins. You put anti-D. Anti-D can work as a positive control. After doing that, you add equal volumes of anti-human globulin. So in every tube, you're going to put other 2 drops of, here 2 drops, here 2 drops of anti-human globulin reagent. Then you intubate at 37 for like 5 minutes. So after adding, you mix. We have added, then we mix. After mixing, intubate. And after intubation at 37 degrees centigrade, that's when we centrifuge. And we said we centrifuge at 1,000 revolutions per minute for 1 minute. After we have centrifuged, you tap to suspend the cells, then you examine for agglutination, for the presence of agglutination of visible clamping. And how do you examine this agglutination? This agglutination can be examined by just looking at the presence of clamps in the tube, or by using a microscope under ×10 and ×40. So we can do macro, that is by using our eyes, by tilting the tube to see presence of clots, or we can do microscopy under ×10 and ×40 to observe for the presence of agglutination. And when we find there is agglutination, it means, so results, the report results is presence of agglutination or clamping. It means it is the DAT or direct Coombs test positive. And no agglutination, it means it is DAT negative. And remember to always include positive and negative controls for the validity of your test. So this is what we can call direct Coombs test, or we call direct anti-human globulin, which detects the already sensitized cells in the body. And we want to see how is it different from indirect Coombs as we are finishing. For indirect Coombs test, hope you are following very well with us. And please, we always urge you to subscribe if you haven't. And then you can also become a member of our channel by subscribing 10,000 per month so that we can continue to grow. So we are seeing under indirect with indirect Coombs. With indirect anti-human globulin test, which we abbreviate as IAT. This is different from direct. How? This one, it detects presence of incomplete antibodies, that is always IgG type, or complement saturating in serum, which is different from direct, which is looking for already sensitized. This one is detecting saturating incomplete antibodies or complement in the serum of the patient. So that is how it differs. So meaning these cells are not yet sensitized in the. So we are supposed. So here under indirect, sensitization is done in vitro. Sensitization, or what we call coating of cells, is done in vitro. That is in a test tube. So you are collecting blood from the patient without coated cells. And how do we do it? We are going to see that if I have serum, this is serum. This serum is from maybe the recipient. This one is majorly applied in routine cross-matching. So if this serum is from the recipient, then you go ahead and add 5% RBC, 5% donor cells. These are donor cells, washed donor cells. We always use washed 3 times. So if we use these 5% donor cells, these cells, they are not yet coated. So after mixing them in one tube, these ones are mixed in a tube, we are going to intubate. We intubate at 37 degrees centigrade and at 37 degrees for 30 to 60. We can use 30 to 60 minutes. This is the intubation. Why are we intubating? We are allowing, in the case this serum of the patient, if it contains incomplete antibodies or the complement, it is going to allow sensitization of these cells in the test tube. So we are going to see if these cells are present in the test tube, they are going to be sensitized. So we shall have our red cells from the donor, and they are sensitized during intubation. These incomplete antibodies come and attach on the surface. They come and attach on the surface, but without causing visible clumping because they are small in size. So these red cells become sensitized. That's why we said sensitization is done in vitro in test tube. After sensitizing them, they do not, but with here there is no visible agglutination, no visible clumping, because these are incomplete antibodies. They cannot cause visible clumping. So after intubation for 30 to 60 minutes, then that's when we again wash with normal saline. After that, we come and wash. So after intubation, we wash with normal saline 3 times. After washing, that's when anti-human globulin reagent is added. So we come and add After washing, we come and add anti-human globulin. We come and add anti-human globulin. This is anti-human globulin is added after washing, and when it is added, we are going to find out that our red cells which were sensitized in the test tube, if we had our red cells which are sensitized in the test tube, after being sensitized by these incomplete antibodies, they are sensitized. Then on addition of anti-human globulin, it is going to cause cross-linkage. This one is going to produce cross-linkage. It is going to cross-link these incomplete antibodies by binding of the FC portion. It causes cross-linkage, and this cross-linkage is the one that brings about visible agglutination, visible clamping, what we call agglutination. So this is what we call indirect Coombs, which is different from direct Coombs. We saw in indirect Coombs, sensitization or coating of red cells is done in the test tube, that is in vitro. How? By getting the recipient serum containing incomplete antibodies, intubating them at 37 for 30 to 60 minutes in the presence of donor washed 5% donor washed red cells. And during intubation, these cells cause sensitization, and after sensitization, without producing visible clamping. So after washing, we first wash, then we add Coombs reagent. And this Coombs reagent is able to bring visible clamping or agglutination by producing close linkage in between these incomplete antibodies after attaching on the FC portion of the incomplete antibodies, hence producing visible agglutination. So this is what we can discuss with you in this session, where we have seen both indirect and direct anti-globulin test. The procedure, the difference in the procedure is that here we are using already sensitized cells, but here we are sensitizing in the tube. So among the procedure, you're going to prepare 5%. How? By getting 1 drop of the red cell, washed red cells, and mixing it with 19 drops of 0.9 normal saline. After forming these donor cells, you add them to the serum from the recipient. If this serum from the recipient contains incomplete antibodies or the complement, during intubation, they are going to sensitize the cells. So after intubation, you're going to wash 3 times, and after washing, you add equal volume of anti-human globulin reagent. And after adding it, it is going to cause cross-linkage in between, producing visible agglutination. And this visible agglutination is examined macroscopically by tilting the tube, examining the presence of clamps within the tube, or you get a pipette and you put on a slide and examine microscopically under ×10 and ×40 objective lens to rule out agglutination. Presence of agglutination on the results, it implies there is presence of incomplete antibodies in the serum of the patient, which are directed against the red cells. Then no agglutination in direct anti-human globulin test is negative. And where do we apply this? This indirect Coombs test is applied in cross-match, routine cross-match. It is also used throughout weak DU antibodies. We can also use it to demonstrate rare antibodies of rare blood groups like K, like M and N, and Lewis. Then we can also use it to demonstrate RBC antibodies circulating in the serum. So this is, we saw direct is used for ruling out hemolytic disease of the newborn, autoimmune hemolytic reactions, blood transfusion incompatibility reactions, and drug-induced hemolytic anemias. But this one is for routine cross-match to rule out saturating incomplete antibodies or complement, to rule out weak DU, that is rhesus weak, someone with weak D. And also we can use it for demonstrating antibodies of rare blood groups like Lewis, like K and M&N. Then we can also use it to rule out RBC antibodies, both applied in blood transfusion and immunology, to rule out these reactions. So thank you so much for learning with us, whereby we have looked at many things. We have looked at production of anti-human globulin by injecting animals with human globulins, then collecting polyclonal or monoclonal antibodies. And we have seen later, we saw the purpose is to demonstrate incomplete antibodies or complement, which sensitize cells without visible clamping. So we add this anti-human globulin to produce visible clamping. We have seen different forms, that is direct Coombs and indirect Coombs, and we have seen the difference. In direct, it is one stage, whereby we're using already sensitized cells in vivo, whereas in direct we are sensitizing cells in the tube that is outside the body. That's the difference we have seen under direct and indirect. And we have seen direct is used for these 4 applications, whereas indirect is for this cross-match, weak DU, then rare antibodies and RBC antibodies, which can cause blood transfusion reactions. Thank you so much for learning with us. Hope you have gained a lot, and may God bless you.